Okay, so tonight we have Dr. Kelly with us again. And for those of you that were around last year, Dr. Kelly gave the first part of this talk where he dealt with the history of chemical warfare agents. And unfortunately, we ran a little short on time and we didn't get into the latter part of this talk. And so this semester, he's agreed to come down and give us the last part of the talk, which deals a lot with neurotoxins. But I think this is kind of a typical of chemistry. There's just so much good stuff in it that you always run short on time, right? It always leaves you wanting more. Now this time we were lucky enough to get a second chance at it. Anyways, I'll turn it over to you.
Thanks a lot, Jason. I appreciate being given the chance to come here again and speak to you again. Jason was one of my favorite students, and I know he's a great instructor. When I was telling my wife the other day, I said, I'm going up to Alva again to give another talk. She said, aren't they tired of hearing you speak yet? I said, not yet, but probably after tonight they will. But as Dr. Wickham said, this is sort of a continuation of a talk that we started last year, right about this time, on chemical warfare agents. Is there anybody in the room who was here last? Okay, so a lot of you have seen sort of the early days of chemical warfare. I'm not going to spend a lot of time talking about that again. We're going to talk about the later events that occurred in the history of chemical warfare when special types of chemical agents called the nerve agents were developed. And this represented a major increase in the lethality of chemical weapons. And again, I always start my talk by telling people why I'm interested in chemical weapons, and that's because a little bit less than 30 years ago, when I was employed by Uncle Sam wearing a green uniform, I was in the chemical corps, and I was my specialty was in chemical weapons. So let's continue our talk right here. And again, I'm just going to define what is chemical warfare, or what are chemical warfare agents. These are poisons that kill, injure, or incapacitate. Oftentimes you want to do any one of the three. Okay? Chemical warfare can be gases, liquids, or dispersed aerosols. Chemical warfare has a considerable history of use in conventional and unconventional areas. In conventional warfare, as we talked about last time, World War I introduced the large-scale use of chemical weapons with at least 100,000 deaths during that war as a result of chemical weapons. Unconventional warfare, terrorists. Maybe some of you are aware that in the 1990s, a Japanese terrorist organization generated a nerve agent called Sarin and released Sarin in the Tokyo subway as part of just sort of a random terror act. And that killed, I believe, 3 people. Okay? So chemical weapons are still being used. So just a little bit of a discussion. What are the different types of chemical agents that are used? Well, there are harassing agents, and the harassing agents are things like tear gases, which you're probably familiar with from popular culture. And tear gases, as the name implies, it causes severe stinging of the eyes and temporary blindness. These are chemicals designed to temporarily incapacitate somebody. Right? So police officers carry mace. Some states allow the public to carry tear gas, such as mace. Okay? These are not lethal chemicals, although if you get sprayed in the face, sometimes you wish they were. And another type of harassing agent, non-lethal agent, are vomit gases. Now, the lethal gases, the lethal agents, these were the agents that were introduced in 1914, in the early stages of World War I, and these are highly toxic chemicals whose only purpose is to either kill or permanently incapacitate. All right? And of the different types of lethal agents, there are what we call the asphyxiating or choking gases. These are gases like chlorine gas, phosgene gas. These are designed to irritate the respiratory system. If the respiratory system gets irritated to enough of a degree, water will start to collect in the lungs, cause something called edema, and you literally drown in the fluid in your lungs. Okay? There are other types of agents, blister agents. These are designed to cause massive chemical burns on the skin. There are agents we call blood agents. These are things like hydrogen cyanide. You've probably all heard that hydrogen cyanide is a deadly poison. Okay? This interferes with the body's ability to transport oxygen. Okay? Cyanide actually binds to hemoglobin, the iron in hemoglobin, in place of oxygen. And then finally, what we're going to talk about today are the nerve agents. And the nerve agents are chemicals that interfere with the body's nervous system, prevents the transmission of impulses from the nerves to the muscles. And these are typically compounds which are organophosphate compounds. They contain phosphorus and other organic functional groups. And they act by being what we call acetylcholinesterase inhibitors. And we'll talk about exactly how they work in just a little bit. Quick timeline. We're going to be starting here, where we're going to talk about the first generation nerve agents, and then here, which are the second generation nerve agents. This is in the 1930s to the 1950s. This is the 1950s to the 1970s. Okay? So the question is, why do you want to use chemical weapons like nerve agents? And I think it's pretty much summed up by this statement here. Chemical weapons are a kind of poor man's atomic bomb. Right? This was made by an Iranian premier back in the 1990s. And what that means is that chemical weapons can inflict massive casualties. They're weapons of mass destruction, and they're relatively cheap, especially compared to nuclear weapons. And one of the things that the military does is it sort of rates the lethality of a weapon or a weapon system. And they use the English longbow that was in common use in the 14th century as their reference point. And we can see as weapon technology developed, the lethality increased significantly. And what I want to show here is in yellow, a nerve gas attack against an unprepared populace or a group of people is among the most lethal types of weapons that are available right now. It has a lethality index of 100,000. The only thing that's larger is a 1 megaton strategic nuclear device, which has a lethality index of 1 million. Okay? So they're very lethal. And I have some information here about how much nerve agent has been produced by the United States in the last 40 years, and the relative cost of the nerve agent. And all of the nerve agent that's been produced, which is enough to kill the population of the planet, okay, has had a total cost of, what is that? About, I can't make it out. I could read this one right here. It's about $600 million. Not even $1 billion. Okay? So as the people in the military are typically say, it's not so much that you get a lot of bang for your buck because nothing's exploding when you have chemical weapons, but you're getting a lot of death for your dollar here with chemical weapons. Okay? So let's now look at the history of nerve agents. Well, the first nerve agent that was actually converted into a weapon of war, a chemical weapon, was discovered accidentally in Germany in the 1930s. There was a German chemical company by the name of IG Farben. And IG Farben was a famous chemical company, and they were the world's leading producer of pesticides. in the 1920s and 1930s, it was discovered that you could kill bugs, especially bugs that were eating your food and your crops, by the use of pesticides. And IG Farben was a major pesticide producer. And one of the chemists working at IG Farben on developing new pesticides went by the name of Gerhard Schrader, right here. Well, in January of 1936, he had a lab accident. And one of the chemicals that he was working on, one small drop of that chemical spilled onto his lab bench. And that one small drop, either through inhalation of the vapors or maybe they came in contact with that one small drop, was enough to sicken Gerhard Schrader and his lab assistant and put them in the hospital for 3 weeks. Okay? They took that chemical that they were working on and they tried to turn it into a pesticide, but it was found to be too toxic to humans to be an effective pesticide. You want to kill bugs, you don't want to kill the workers in the fields. Okay? Well, that compound that was discovered by accident, or the toxicity of the compound was discovered by accident, is this chemical substance right here. It is an organophosphate. There's our phosphorus. Okay? This compound goes by the name of ethyl N,N-dimethyl phosphoramido, because it has a phosphoramido group, cyanidate, has a cyanide group right there. So if you've taken organic chemistry, hopefully you can sort of understand the nomenclature of this compound. Well, it was given the trade name Tabun. When they were trying to market it as a pesticide, it was given the trade name of Tabun. Okay? Now, Tabun also has another designator. This is a U.S. military designator. This is called Agent GA. We're going to see a number of nerve agents that are going to have G as the first letter of their identification. That stands for German. Tells us that they were first prepared by the Germans. Okay? Now, this compound here, it's a volatile liquid. Okay? So it has a fairly high vapor pressure. And it will cause toxicity either through inhalation of the vapor, or it's actually absorbed through the skin as well. This passes through the skin relatively easily. Now, when you're exposed to Tabun, or Agent GA, it has a number of effects. Now, these effects come about because of inhibition of the nervous system. It affects the nervous system. Specifically, it's going to affect transmission of information from the nerves to the muscles. Okay? So when you look at a lot of these effects, they come about because of this inhibition. So it tends to cause nervousness and restlessness. It causes miosis, contracted pupils. It causes rhinorrhea, which is a runny nose. It causes excessive salivation, difficulty swallowing, difficulty breathing, excessive sweating. It causes bradycardia, which is a slow heartbeat. It slows the heartbeat. It causes loss of bladder and bowel control. It causes apnea, which is stopped breathing. Ultimately, loss of consciousness and death. You have this symphony of symptoms that sort of cascade down from the top to the bottom as this chemical Tabun does what it does. How about a little chemistry here? So I'm going to assume there's a lot of people who've done organic chemistry. There's some relatively simple organic chemistry to make Tabun. It's a simple synthesis overall. You start off with this compound right here, phosphorus oxytrichloride, or phosphorochloride. If you've taken organic, you might recognize that we use this reagent to bring about dehydration of primary alcohols. Okay? And we're going to take secondary amine here. We mix them together. We're going to form this compound right here, which is a dichlorophosphoromide. Dichlorophosphoromide. Okay? We then take the dichlorophosphoromide. We're going to add 2 equivalents of sodium cyanide, 1 equivalent of ethanol, and we get a reaction, and that makes the ethyl N,N-dimethyl phosphoramidocyanidate. There's tabun right there. What? 1, 2 steps. 2-step synthesis. So we see that tabun is easy to produce, except for the fact that when you produce it, how do you handle this stuff? It's really, really deadly. Okay? So that's really the only limiting factor to the preparation of tabun. Now, when you try to make tabun and store it, you've got a problem because tabun is very corrosive. Okay? It will corrode stainless steel, so it's difficult to store and stockpile. Okay, now, for reasons I'll discuss in a minute, the Soviet Union, back in the Cold War, this was their major chemical agent, tabun. And they stored weapons that contained thousands of tons of tabun. And I've seen pictures of their old shells and bombs in storage centers today. And tabun has corroded away the casings of the shells and bombs, and they have storage centers where tabun is leaking out probably by the deciliter. Okay? So you want to talk about hazardous waste. That's about as hazardous a waste as you can get. So it's pretty corrosive. It's very difficult to store. That's one of the things that makes it a non-ideal chemical weapon. But the Nazis, during World War II, recognized that this would be an effective weapon. So they developed a plant in 1939, which was completed in 1942, to make tabun. And it had a production capacity of about 1,000 tons per month. And they produced about 12,500 tons during World War II. Now, they produced it but never used it. Okay? And they never used it because they thought the Allies, the United States, Great Britain, had similar chemical weapons, which we didn't. Okay? So it's a good thing that they weren't aware of the situation. It's apparent that the Nazis probably would have used it if they thought there would be no retaliation in kind. Okay? And so this chemical plant tended to be in the eastern part of Germany, so when the Soviet Union overran Germany at the end of World War II, they collected all of the machinery in the plant, including all of the tabun that was stockpiled, and they brought it back to the Soviet Union. And as I said, tabun became the standard Soviet chemical agent during the Cold War. Now, after the development of tabun, there were even more deadly chemical weapons, chemical agents that were developed. Okay? And one of them is this compound right here, which goes by the name of sarin, S-A-R-I-N, and its agent name is GB. Now, sarin is also an organophosphorus compound. This is an this is a phosphono compound. Remember, general chemistry, you're learning about the oxides of phosphorus. Okay, phosphono is the second highest oxidation state. And this is isopropyl methyl phosphonofluoridate. Okay, there's your isopropyl, there's your methyl. The phosphono group means we have 3 electronegative elements connected to the phosphorus, and there's the fluorine. That makes it the fluoridate. Okay? Now, if you remember general chemistry, you remember phosphorus, when it expands its octet, becomes chiral. Okay? And again, organic chemistry, if we wanted to look at the absolute configuration of sarin, the absolute configuration is the S enantiomer. Okay, this is the really active form of sarin. Now, you also have the R enantiomer, and usually for most biological systems, only one enantiomer has activity, the other one doesn't. But this is a situation where both of them have activity, it's just that the S enantiomer has the highest activity. Okay? That has this structure right here, the orientation of the bond. Again, very easy to make. We're going to take phosphorus trichloride, we're going to add methanol, and then sodium and some chloroform, and then PCl5, and there's a precursor to it. Then we treat it with sodium fluoride in isopropyl alcohol, and we get simple substitutions. These are like nucleophilic acyl substitutions on an acyl group. Okay, if you're familiar with acyl chlorides, you know these are very similar to a nucleophilic substitution on an acyl chloride, and we make sarin right here. Okay, now the name sarin comes from. It's interesting how they got the name sarin. It's named after the chemists who first prepared it, and they were Schrader, Gerhard Schrader. He was the discoverer of tabun, and then Ambrose, Rudiger, and Linde. Okay, so they took S-A-R-I-N, they called it sarin. This is the most versatile class of the G nerve agents. It's versatile because it's the most lethal by inhalation, and it's not nearly as persistent as tabun. Okay, what do I mean by not nearly as persistent? Well, these compounds can react with water. They can be hydrolyzed by water. And as we know, water is in the atmosphere, water is in the environment. And so oftentimes, the nerve agent will be essentially affected by any water that's present. Okay? It will decay in the presence of water. Okay? So this is very easily hydrolyzed. So it doesn't persist very long in the environment relative to tabun or to the next reagent we're going to talk about. So why would that be important? Well, if you're going to use it as a weapon, you want to expose the enemy troops to the nerve agent. You want to kill all the enemy troops, and then you want to move through the area where the enemy troops were, and you don't want the nerve agent to be persistent in that area as your own forces are passing through. So that's an important characteristic when we're talking about nerve agents, is its persistence. Sometimes you want it to be very persistent, sometimes you want it to decompose very easily, to hydrolyze very easily, and it's non-persistent. Well, this was discovered relatively late, going into World War II. The Germans really didn't have much of a chance to begin the development of this nerve agent. They built one small pilot plant in 1944. They produced approximately 100 tons of sarin at that pilot plant. 500 kilograms of sarin, 500 kilograms isn't a whole lot, but 500 kilograms of sarin was captured by the Americans at the end of World War II, and sarin, codenamed GB, became the first primary nerve agent to be stockpiled by the United States. Okay, so this was the first nerve agent that America started to produce and stockpile. Okay, then finally, a third member of this class was discovered, or developed, I should say, and it's this compound right here. Now, this is simply an analog of this. Replace the isopropyl group here with this structure right here. We call this a pinacolyl group. Okay, this pinacolyl group. And you have a new nerve agent called Soman, which has the designation of GD. Again, synthesis of this is pretty simple. Okay? And Soman is a little bit different than sarin. And I'm going to tell you that Soman is more persistent, okay? And it's more persistent because of the presence of this group right here. Because you have that group, hydrolysis. Hydrolysis occurs when water comes in and we essentially displace the fluoride. We make a phosphonic acid. Okay, we displace the fluoride. And you need the fluorides here for it to act as the nerve agent. So here's a general question for anybody in the class. This compound does not hydrolyze nearly as fast as this one. For hydrolysis to occur, water has to come in and attack at the phosphorus. Why do you think this is much more resistant to hydrolysis? Anybody want to answer that question? 2 words. One begins with S, the other one begins with H.
Steric hindrance.
Steric hindrance. Right. What we see is this pinakyl allele group really increases the steric hindrance, makes it much more difficult for the water to come in and react. Okay? So this is Soman right here. Now, this is as lethal as sarin, as you might expect, because they're pretty similar structures. They have a similar lethality, but this one is more persistent. Okay? Now, why would you want a nerve agent that's more persistent, that sticks around longer? Well, if you want to prevent opposing forces, enemy forces, from occupying an area, maybe there's a strategic valley that you don't want them to get into, okay? You would then sort of spread this more persistent nerve agent in that area, and that way they're not going to move in there because they don't want to be exposed to the nerve agent. Okay? Now, Soman has not been effectively used by either the United States or the Soviet Union during the Cold War. And we'll explain why in a minute. But remember I said that Germans had all of this tabun, but they never used the tabun because they were afraid that the Allies could retaliate in kind. Okay? They were afraid of that. And the Allies weren't totally ignorant of the chemical agents that the Germans had. They just hadn't been able to develop any chemical agents as effective. Okay? The British in particular were aware of the German research on these organophosphorus-containing insecticides. In fact, Schrader had published a paper on this compound right here and its potential as a very powerful insecticide. And there were some British chemists who worked for the British military, read that paper, and realized that this class of compound could be a very effective chemical warfare agent. So the British started their own research on chemical agents, and they made this compound right here, which is diisopropyl phosphorophoridate. Okay? And this is called DFP. Well, DFP does act as a nerve agent, has all of the same properties as GA, GB, and GD. It's just not as good. Okay, it was not as effective. This little table here compares the lethal dose required to kill. Okay, here's DFP. This is toxicity by injection into mice. These numbers represent the dose in milligrams per kilogram of body weight. And you can see that DFP is about an order of magnitude less effective than the other chemical agents. Still effective, just not as effective. So the Allies did understand these chemical agents. They were just a little later to the game and didn't have a chance to develop them as much. But after the war, things changed. All right? And the Cold War started. The United States and Great Britain knew that the Soviets had taken thousands of tons of tabun to Russia and were actively working on weaponizing these things. So they started their own chemical warfare research. Okay, whoops, I didn't change it. Went the wrong way. So let's talk a little bit about post-war research, and now these are the second generation agents, which are given the prefix V. Okay? And again, these were discovered accidentally. Again, there were chemists who were working on the development of pesticides. Okay, after World War II, the population of the world started to increase at a great rate. There was a need to grow more food in developing countries. Pests are a big problem. There was attempts to sort of prepare new effective pesticides. And a British company, Imperial Chemical Industries, developed a pesticide called Amaton. This is Amaton right here. And we can see that Amaton is an organophosphate type of compound. Okay? The actual chemical name of Amaton is OO-diethyl. Okay, there's two ethyl groups right there. S2-diethylamino ethyl. Diethyl amino ethyl. Phosphothiolate. Thiolate means the sulfur. So that's its chemical name right there. It had a train name of Amaton. Amaton was actually produced and marketed, okay? But it was very quickly taken off the market because it was too toxic to use in agricultural production. In the space of a short period of time, there were 17 deaths attributed to the use of Amaton. Its toxicity is about equivalent to sarin. Okay? Now, the chemist who synthesized Amaton, his name was Ranjit Ghosh. Okay, he was an Indian chemist living in Great Britain at the time. Okay? Amaton is still being made in the world today. There's a treaty, an international treaty. All the countries of the world signed it, except for North Korea in 1990, which prohibits the manufacture of chemical weapons. Well, Amaton had a little bit of a disclaimer associated with it because, quote, it is an agricultural product. Countries can manufacture small amounts of it. Okay? And the only country we know of that has taken advantage of the agricultural exemption for Amaton is North Korea, which has stockpiled very large quantities of Amaton for, quote, agricultural export. Okay? They don't seem to be exporting much of it, but they're making a lot of it. Okay? Okay, so we look at the structure of Amaton, and Ranjit Ghosh was then hired by the British military to work in their chemical weapons division. And soon after Ranjit Ghosh began to work at the British research facility in a place called Porton-Down, a new nerve agent was developed, and this is called Agent VX. Okay? Agent VX was super secret. The British were keeping it to themselves. The Americans found out about it, and they wanted to get information about VX. And the Brits said, okay, you trade us information about your thermonuclear weapons, and we'll give you the information about VX. And so the information was transferred to the United States, and the United States then began the large-scale preparation of Agent VX. Agent VX, again, represents a major increase in the lethality of the nerve agents, okay, for reasons which we'll talk about in a little bit. Okay, now this is called Agent VX. What does the V qualifier mean? Remember, the G qualifier meant German. The V qualifier means venomous, all right, which I think is kind of appropriate for Northwestern, since I think you're doing a lot of research on venom. Well, this is not your typical snake venom, but the V stands for venomous. Here's some chemical syntheses that are going on to make VX. This was the initial synthesis right here. You take phosphorus trichloride, you take methyl iodide, you're going to make methyl dichlorophosphine, then you react it with some ethanol and base, and you're going to make the diether here. You react that with phosgene, and you get this compound here, this precursor, which I have in red. This is ethyl methyl phosphenochloride. Okay? But it's in red. Why is it in red? Because this itself is also a very effective nerve agent. So you have an intermediate product on the way to what you want to make, which itself is incredibly toxic. Okay? You then take this intermediate, you treat it with this compound right here, and you make VX. Well, this was the initial synthesis, but this was soon abandoned because you're dealing with an intermediate which itself is very toxic. Okay? So what we have here is the modern synthesis. Okay, and I was partly involved in the modern synthesis when I was in the chemical corps in the Army. But in the modern synthesis, as you work through the synthesis to get to VX, you don't have any particularly toxic compounds. All right, none of the precursors are really toxic, really deadly, until you get to VX. And the key to the modern synthesis is the preparation of this intermediate, which is given the code name QL. Okay? If you take QL and you just mix it with sulfur, okay, that's pretty simple. Sulfur is not particularly toxic. QL is not particularly toxic. If you take it and mix it with sulfur and then heat it to temperatures above 70 degrees, you will form VX. So you don't get your toxic material until the absolute step of the synthesis. And when I was in the chemical corps, we were working on types of munitions called binary munitions, where you have two innocuous chemicals, and when put them in a shell, when the shell is fired, they mix together and they form the nerve agent. We were working on this right here, trying to develop the technology where they would mix together efficiently in an artillery shell. Okay, so that's the synthesis of these drug agents, of these nerve agents. Talk a little bit about their properties, in particular VX. VX is superior to the other nerve agents. Why? It has a lower volatility. Well, you might think, wait a minute, lower volatility, there's not as much nerve agent in the air. Aren't you supposed to inhale the nerve agent? Well, the answer is it's actually better to be exposed to the liquid droplets of the nerve agent than the gaseous form of the nerve agent. So having a low volatility is good. It's stable to hydrolysis, which means it's persistent. These tend to be fairly persistent nerve agents. It's rapidly absorbed through skin and clothing, and stable to hydrolysis again. Much more toxic. It has a much higher lethality. All right, you can look at these numbers right here. There's DFP, that was sort of the British squib, the nerve agent that wasn't quite good enough to be a nerve agent. Tabun, Sarin, Soman, and VX. You can see that VX is, especially in terms of human percutaneous toxicity, much more toxic. Okay? Now, VX is persistent. This little plot here shows us the half-life of VX in terms of time. These are days here. And you notice that VX itself is pretty persistent. It doesn't degrade very quickly. However, if we add base, and I guess this lower axis here is base, we're looking at pH. Under basic conditions, once we get that pH down to about 8, okay, we're getting into basic conditions here, we can then start to hydrolyze VX. Okay? So, again, going back to organic chemistry, remember base hydrolysis of an ester. Right? When you have a base hydroxide, you can hydrolyze an ester fairly quickly. Whereas water, it does not hydrolyze very quickly unless you have an acid catalyst. Okay? So VX is persistent, but it can be degraded at high pHs. All right, let's get to the part that's pretty interesting, and that is, what is the mechanism of nerve agent action? How does it do what it does? Well, it inhibits a key enzyme called acetylcholinesterase. Okay, now acetylcholinesterase is a very important enzyme in your body because it brings about the hydrolysis of a neurotransmitter called acetylcholine. This is acetylcholine right here. As you can see, it's an ester. Okay? It has an acetyl component right here, and it has a choline component right here. In the presence of water, we can hydrolyze this ester, and we get an acid, and we get choline. But then again, how about organic folks? How quickly does the hydrolysis of an ester occur when you just have water present? Is that a fast hydrolysis? It's a particularly slow hydrolysis. All right? It needs to be catalyzed somehow. In the lab, we catalyze it with acid or base. In the body, it's catalyzed by an enzyme. Now, why do you want to take acetylcholine, which is a neurotransmitter, and hydrolyze it or break it down so it's no longer a neurotransmitter? Well, we have to have a little bit of an understanding of what happens when a nerve signal travels from a nerve to a muscle. Okay? So here's our nerve here, here's our muscle cell here. They don't touch one another. There's a space between them. It's called the synaptic gap or the synaptic cleft. And we need to get a signal or some sort of information, we're going to use chemical information, from the nerve to the muscle. So what happens is there's an action that occurs, say in the brain, and that action is transmitted by a series of chemical polarizations of the nerve cells. And we get down to this nerve cell right here. Now your nerve cells have these little vesicles that contain acetylcholine. Okay, simple chemical that's synthesized within the, or generated within the cell. And these little vesicles are typically migrate to the cell membrane. And when a chemical signal comes down, it causes these vesicles to release the acetylcholine into the synaptic gap. The acetylcholine then travels across the synaptic gap, and it binds to other types of enzymes which are on the surface of the muscle. These are acetylcholine receptors. And when the acetylcholine receptor binds to the muscle, it causes the muscle to essentially fire. Okay? We have the events that cause muscles to contract once the acetylcholine is bounded to the receptor. Okay? Now, also on the surface of the muscle are other enzymes. This is an acetylcholine esterase. Again, if you've taken biochemistry, you recognize that this is an enzyme that's going to cleave an ester. It's going to hydrolyze an ester. Okay? And why do you want to hydrolyze acetylchlorine? Okay. Who remembers the mass effect from general chemistry? You have an equilibrium. You have an excess on one side. It tends to drive the equilibrium. Right? Mass effect in biochemistry, if you have a substrate that binds to an enzyme, and you have an enzyme, as you increase the concentration of the substrate, you're going to drive that system over into an enzyme substrate bound situation. Okay, that's the mass effect. So when our nerve cell releases all the acetylcholine, there's lots and lots of acetylcholine present, here, and they tend to now have a high concentration. They can then bind to the receptor sites that are going to cause the various activities in the muscle. There are two types of receptor sites. There's the muscarinic receptor site. We see these typically in smooth muscles and glandular systems. And then there's the nicotinic receptor site. We see these typically in skeletal muscles. Okay, so you've got a high concentration of acetylcholine. You're going to drive your equilibrium into the bound state. You're going to fire off your muscle. Once the muscle has fired, or once you have a high equilibrium of the receptor-bound system, you want to then begin to move the equilibrium in the other direction. Otherwise, the muscle is just going to sit there and it's just going to keep firing. Okay? And so you need to begin to move the equilibrium in the reverse direction. You want to unbind the substrate from the receptor. Well, to do that, you have to start getting rid of some of the acetylcholine. You need to reverse the mass effect. And that's where acetylcholine esterase comes into play. When you have acetylcholine esterase, all of that excess acetylcholine that's present in the synaptic cleft starts to get hydrolyzed by acetylcholine esterase. Okay? So you quickly start to chew up all of the excess acetylcholine here, and you then reverse the equilibrium and you unbind your receptor sites. Okay? Well, acetylcholine esterase is the fastest hydrolytic enzyme known. It works incredibly fast. It has an incredibly high turnover rate, which you have to have because you have to start immediately reducing all of that excess concentration of acetylcholine to get back to your normal muscle state. Okay? You have to regenerate the normal muscle state. Now, if you block acetylcholine esterase, you maintain this high concentration of acetylcholine, and your muscle is always now in its excited state, contracted state, and that's not good. Essentially, the muscle then ceases to function normally. It seizes up. Okay? And that's sort of what you're seeing when you have a chemical agent. All of the nerve agents, the organophosphate nerve agents, block acetylcholine esterase. They prevent it from reducing the concentration of the acetylcholine. What we see is that our receptor sites on the muscle are bound, and the muscle continues to fire. That's not a good thing. So here's a little model here. This is a model of acetylcholine esterase activity. When we look at acetylcholine esterase, and we're going to look at a picture of it, an X-ray structure of it in a minute, we define two specific regions of the ester, okay, which are necessary for the activity of acetylcholine esterase. There is what we call an anionic site in the ester. Anionic site, just as its name implies, it's a region of negative charge or rich electron density within the ester. Okay? And then there is another site, which is the esteratic site. This is the site where the chemical hydrolysis is going to occur, and one key amino acid residue in the esteratic site is a serine. Okay? Serine has an OH group. Okay? So what happens here is, this is acetylcholine here. Acetylcholine is a quaternary ammonium nitrogen. It has a positive charge. That quaternary ammonium nitrogen is attracted to the anionic site, right? Positive-negative attraction. What that does is it brings the ester portion of acetylcholine close into the esteratic site, where the serine, with its hydroxyl group, now begins to react with the carbonyl of the ester, okay? And we are going to start the hydrolysis. Well, this is a simple model of the hydrolysis. We're just trying to show you that the serine now, the serine hydroxyl group is going to get involved. Okay, it's going to help the cleavage of this carbon-oxygen bond. This part goes away as choline. We initially get the acetyl group bound to the serine oxygen. We form a new ester. We've taken one ester and formed the new ester. And then water comes in, hydrolyzes the acetyl group, gives us acetic acid. Okay? And regenerates our acetylchlorine. So this is sort of a rough picture of what has to take place for acetylcholine to do its thing. The important residue, people who haven't taken biochemistry yet, when I talk about a residue, that means a specific amino acid in the peptide sequence of the ester. Okay? And so this is a serine amino acid, which is part of the sequence. Okay. So this is really cool. When I first started learning about acetylcholine esterase, we knew nothing at all about what it looked like. And acetylcholine esterase was one of the first biological proteins to get its picture taken, to get an X-ray structure of it. And the reason why it was one of the first to get its X-ray crystal structure determined was because the Department of Defense paid mucho dollars to crystallographers to get a picture of what this looks like, right? When you're doing chemical warfare work, and it's dependent upon acetylcholine esterase. You want to look and see what it looks like. So this is a crystal structure. Okay. Now, when you're looking at a crystal structure, you see all of these little wiggly lines. Those wiggly lines represent the backbone of the enzyme. And biochemists tend to use curlicues, which are helices, to represent the alpha helices that are present in these enzymes, and sort of these ribbon strips that are sideways to one another to represent the beta strands. Again, folks who've had biochemistry will recognize this. Okay? And what we're looking at is the cartoon representation of the backbone, and then superimposed over the cartoon representation is sort of the space-filling model. So we see the shape. Now, the neat thing about acetylcholine esterase is that if you look in right here, what you see is essentially a crater in the structure. This is the active site. And I love to describe acetylcholine esterase as a pitcher's glove. Okay? People who've seen a pitcher's glove will know exactly what we mean. It's a pocket surrounded by the glove. And it's in this pocket here, which is where all of the activity takes place. Now, deep down in the bottom of the pocket is what we call the esteratic site. These contain the residues that are going to be directly involved in the hydrolysis of the ester. These yellow sites here are going to represent the anionic site. These are the regions of negative charge, so that they will hold the acetylcholine in place. Remember, the acetylcholine has a positive charge. Okay? The three key residues in the esteratic site are serine 203, glutamic acid or glutamate 334, and histidine 447. Again, if you haven't taken biochemistry, these numbers here represent their position along the sequence. Okay? The sequence of different amino acids in the peptide, in the protein. Here's the same structure now. I've taken away the space-filling model. And again, there's the crater that you see here. Okay? So that's one picture of the molecule. For people who haven't had biochemistry, these are the different amino acids that are going to be involved in both the esteratic site here and the anionic site. Anionic site contains aspartic acid. Why is it anionic? Because at the pH measured, aspartic acid is in its carboxylate form. Okay? And then other residues that are in the anionic site have these aromatic rings. That's going to create sort of a pi cloud, which is a region of negative charge. Okay? That's going to attract and hold the quaternary ammonium nitrogen, the nitrogen with a positive charge that's present in acetylcholine. And then taking that same crystal structure we saw before and using some visualization software, we can blow it up. And this is the key amino acid residues. There's the serine right there. There's the OH group. Here's a glutamate group. There's the histidine group right there. And then these here are the amino acid residues of the anionic site. Okay, same slide again. Okay, here's the same slide again, showing the amino acid residues. Now, this slide is a slightly different crystal structure of the same enzyme. Okay, but this is a really interesting crystal structure because this right here, where I'm showing right here, that is a molecule of sarin, which has covalently bonded itself to the serine 203 residue. So there's our nerve agent right there. It has bonded itself permanently. We've got a covalent bond to the key active site of our enzyme. Okay, so that's why these compounds inhibit acetylcholine esterase. They completely block acetylcholine esterase from getting in to where it needs to be. Okay, we'll get back to that in a minute. So, if you got, well, I'm running late again, but let's take a look at this slide right here. Just a simple model of the hydrolysis of acetylcholine by acetylcholine esterase. So here's our acetylcholine right here. Here are some of the key amino acid residues. This is the esteratic site right there. Okay, there's our serine with its OH group. These are the anionic site residues. Here's the quaternary ammonium nitrogen. So first, we sort of lock acetylcholine into the site because of the interactions that we see right here. Okay. Then the hydroxyl group of serine reacts with the carbonyl carbon. We simply start to have a nucleophilic acyl substitution process. We generate the tetrahedral intermediate, as we've seen there. Okay. After forming the tetrahedral intermediate, we then lose choline from the structure. All right. So there's choline, it's gone away. Now we have generated this ester right here. Choline leaves, and now water comes in. And water now is involved in the hydrolysis of this ester. Okay? We need to hydrolyze this ester away to get it off of the serine 203 so we can go again. Okay? And this is where the water comes through. And it hydrolyzes the ester. It gives us the acid, in which case it happens to be acetic acid, and the acetic acid goes away. Now, what does the nerve agent do? The nerve agent takes the place of acetylcholine, binds itself permanently into the serine. That is a very difficult to hydrolyze covalent bond. It is a permanent bond. Okay? Okay, so this just sort of summarizes now what the nerve agent does. Here's our nerve agent here. There might be some small attraction between our groups on the nerve agent and the anionic side, but now the nerve agent comes in, it reacts with the serine, okay? And the serine now forms a permanent covalent bond to the phosphorus. Those oxygen-phosphorus bonds are pretty stable, okay? Very difficult to hydrolyze. We have now effectively killed acetylcholine esterase. If you kill acetylcholine esterase, you can't affect the concentration of acetylcholine that's in the synaptic cleft. The muscle keeps firing, you stiffen up, and then all sorts of bad things happen, okay? Again, here's our crystal structure showing, this is sarin right here, showing sarin, which is covalently bound to the serine residue. Okay, if you just bear with me, I can run through this in about another 5 minutes. So, is there any kind of antidote to nerve agent poisoning? Is there any way we can prevent nerve agent poisoning? And the answer is yes. Okay? We can prevent nerve agent poisoning, and we can also have an antidote if nerve agent poisoning occurs. And there are a number of chemicals that are acetylcholine esterase protectors. All right? They will protect the acetylcholine esterase from the nerve agent. They're effectively going to block the nerve agent from getting into that pocket that's present in acetylcholine. And they do so by actually binding reversibly to the acetylcholine esterase. So this is kind of interesting. You have a protective chemical substance which itself is an inhibitor of acetylcholine esterase. It competes with acetylcholine for the acetylcholine esterase site, but because it's a reversible inhibition, it actually protects the acetylcholine esterase. Okay? And it works something like this. We have compounds, these are carbamates. neostigmine, okay, and pyridostigmine, okay, and we can see that they contain either an aromatic ring with a quaternary site or a pyridine with a cationic site next to a amide functional group. Okay, now these are reversible competitive inhibitors of acetylcholine, which means if you have them present, they can come in and they can react with the esteratic site and they can sort of form a temporary inhibited complex here. Now that's just temporary. That's the important thing. But while these carbamate protectors are within the active site of this acetylcholine, they just physically prevent the chemical agents, the nerve agents, from getting into the active site of the ester and reacting at the active site of the ester. Okay? So typically, if you expect to be exposed to nerve agents, these would be given before the exposure as a prophylactic. They don't work at all after the fact. Okay? If you've been exposed to the nerve agent, these don't work. But if you haven't been exposed and you get a significant dose of these protecting agents, they will prevent the nerve agent from permanently binding to the active site of the ester. Okay, there are other things we can do as well. Remember, the whole problem is that when you have a high concentration of acetylcholine, it remains bound to the muscle receptors. So if we had another chemical substance that could block the muscle receptors, okay, that would prevent the muscles from continuously firing. That's what kills you, is the fact that the muscles continuously fire. And so we have a series of what we call anticholinergic drugs, and these compounds simply block the receptor sites at either the muscarinic receptor site of the muscle or the nicotinic receptor site of the muscle. They keep acetylcholine from getting in there and causing the muscles to fire. Okay, so this is another way we can protect ourselves against the effects of the nerve agent. All right. Now, the best thing would be to have a chemical substance that could actually remove the nerve agent from the serine residue of the ester. Okay? And this is a true antidote. It's going to return the acetylcholine esterase to its normal form. And there is a true antidote to nerve agent poisoning. It is this compound right here. This is called, it has the name pralidoxime, okay, or pyridine aldoxamine methyl chloride, okay? It's oftentimes abbreviated as PAM, 2-pyridine aldoxime methyl chloride, or PAM. Now, PAM now is used by the U.S. military and other service forces around the world as a true antidote to nerve agent poisoning. They have little injectors. It's a kit. If you start to detect the symptoms of nerve agent poisoning, you just stick the needle from this kit in a thigh and injects the chemicals into your body. Now what happens is this compound right here loses a hydrogen right here. And once it loses this hydrogen, you have a negative charge on this oxygen. And that makes this oxygen what we call a supernucleophile. Because it's next to a nitrogen, which has a lone pair, this oxygen becomes a supernucleophile. And being a supernucleophile, it is now nucleophilic enough that it can react with the phosphorus atom of the bound organophosphate nerve agent. Okay, so we can see a picture of it right here. This is what happens. Here is our bound nerve agent. Again, it's pretty much of a permanent covalent bond. It's hard to displace the phosphorus from this oxygen. Here's our PAM. Okay, the PAM deprotonates at the oxine position. All right, this oxygen now becomes supernucleophilic. It's a very, very strong nucleophile. It's able to come in here. It's able to add directly to the phosphorus. Okay? Forms a bond to the phosphorus, frees up the serine hydroxyl residue, and we're safe at that point. Okay? That is the true antidote to nerve agent poisoning. Pralidoxine or PAM. Okay? And here's some information about how we can treat nerve agent poisoning. We have atropine. Atropine is a nicotinic or muscarinic receptor antagonist. We have PAM, and here's DFP. Here's our nerve agent, right? So we give mice the nerve agent. There's 0% survival, right? It kills them all. We give mice even more nerve agent with just the atropine, and atropine alone is not enough. It kills them all. Okay? We give the mice nerve agent, and now we give it PAM, and there's survival. There's about 22% survival. And then finally, we give the mice nerve agent plus PAM plus atropine, and there's 100% survival. Right? So, at least in terms of the military, which are equipped to handle chemical weapons are not nearly as terrifying as they have been in the past because of the antidotes that are available for them. Okay? Well, I went a little bit longer than I thought I would. Pretty much finished right here. I'm open up for questions if anybody has any questions. No questions.
Anyone have questions? No? Okay, well let's give Dr. Kelly a hand then.